Welcome everyone to our lecture 17, part 2. If you haven't yet watched the BIS103 map part 5 for this lecture, as well as our part 1, the first video where we discuss the organ-specific reactions in the context of metabolic integration, I'd encourage you to do that first because it's really an important foundation for this video where we want to actually apply what we learned about integrating organ-specific and common metabolic pathways under different conditions. And we want to do so by focusing on metabolism in mammals. Right? And we want to look at some maybe extreme conditions in terms of energy resources. So let's start right away with the first one. Let's assume we're in the well-fed state, right? So you just had a big meal, you really fed, you have had lots of calories, lots of amino acids, lots of fats, and lots of carbohydrates come in, and you want to deal with it, right? Under those conditions, right, your feast hormone, insulin, is coming in. And so remember, right, from our lectures on metabolic regulation, what insulin does, right, our discussions on hormones. And again, I didn't ask you to really memorize the exact pathway that insulin is using. If you remember and understand the general concepts of these different reactions and how different pathways are regulated here, especially those that are highlighted, that will be perfectly sufficient for our course. But right, in this state, we're in the well-fed state. We have a lot of glucose, so high blood glucose is coming in. That increases your insulin concentration, and then insulin, much like the other hormones, will trigger changes in several of the opposing pathways, especially here focusing on the breakdown and synthesis of glycogen. Right? So if insulin is increasing, it is triggering this PKB pathway, so a protein kinase B pathway, that will be increased in response to insulin. What it will actually do, it will phosphorylate your glycogen synthase. So the arrows here up would mean upregulation, the arrow down is downregulation. So phosphorylation here increases the activity of glycogen synthase. So you're increasing the biosynthesis of glycogen, which makes sense, right? You have a lot of blood glucose. You can't all use it up for chemical energy production through cellular respiration. So you're putting it away for later. In this case, you're using glycogen that's triggered by insulin. At the same time, insulin is undoing everything that our glucagon, low glucose sensor hormone has done. And what it will do is actually it will use a similar kinase pathway that we didn't discuss, so you don't need to know how it works. But what it will do is it will actually decrease the activity of this phosphorylase kinase that we had said was really important in the regulation of glycogen phosphorylase, the enzyme that is relevant for the breakdown of glycogen. So through this kinase and the downregulation of phosphorylase, insulin is downregulating glycogen breakdown. It is increasing glycogen production so that you can store all those resources away for later. There are actually also a number of other pathways here that insulin is regulating. We haven't touched on them. Just sort of out of interest, you don't have to memorize it. There are actually also transporters here that are increased in their abundance, which allows you to actually take up more glucose by the cell that then can be transferred into storage. So now that we know, okay, glucose is a big part here, right? It's triggering the changes in metabolic pathway directionality. What does this look like in the big picture, right? Again, you had this big meal, you have all the amino acids, proteins, lipids coming in, and sugars. And so this illustrates actually where some of the major pathways are going now in this situation. So again, right, we just talked about insulin is triggering a lot of this. Right, your glucose is high, it's going into the liver. Through insulin-mediated path regulation, you are triggering the formation of glycogen. You're releasing some free glucose because, right, the brain wants always glucose, so some will still be released. And some part will be broken down through your glycolytic pathway into acetyl-CoA and will go into tags, right? In this case, we're not running a lot of ETC because we have so much energy already. So in this case, you can actually also break down glucose through the glycolytic pathway, the TCA cycle, and then you can put it into tags. and they will be stored in your adipose. That's your fat, right? Stored for later. Your amino acids that are coming in here, right? They will be broken down. They can be put into protein synthesis, for example, but a lot of them will be broken down to alpha keto acids, right? They can also be used for production of fats. That's not indicated here, but they can be used to generate fats too. And remember, right, you're dealing also with your ammonium here, right? If you're breaking down amino acids, the free ammonium has to be secreted away from your system. And so that's our urea cycle. So if you have a meal that's really protein rich, your urea cycle would be increased too, right? Because you need to deal with the free ammonium from the amino acid breakdown. So general trend here, you're using all these pathways in an anabolic fashion for the most part here to store away these energy-rich metabolites for later. Now say you haven't eaten for a while, right? You're sitting in front of the videos here, watching them. You haven't really eaten in the meantime. So now your energy resources are starting to drop, right? What is happening now? Now, right, insulin, your high glucose sensor hormone, is gradually replaced by glucagon, the hormone that is sensing lower blood glucose levels. So your blood glucose levels are dropping. Glucagon is increasing. And so what it does is, right, this is the entire pathway we discussed in our lecture on metabolic regulation through the increase of cyclic AMP production, right? And cyclic AMP was an allosteric regulator, an activator of our PKA, or protein kinase A, right? Remember, this was a protein kinase that has many proteins as its substrate and can phosphorylate those. And through this covalent modification of phosphorylation, change their activity. And so it does multiple things, right? The first thing it does is that it phosphorylates the secondary kinase, right? Remember this idea of using multiple kinases to have this cascade effect of phosphorylation, and through activation of a pathway much faster by using cascades of kinases. So here phosphorylation activates the secondary kinase. That kinase, its protein substrate is your glycogen phosphorylase, so the enzyme relevant for glycogen breakdown. That is activated through phosphorylation. So as an end result, you are increasing glycogen breakdown, right? Your blood glucose levels are dropping, glucagon comes in, and it increases release of free glucose now by breaking down glycogen. At the same time, we want to have reciprocal regulation, right? So we want to downregulate now the synthesis of glycogen, right? We're breaking it down more. We want to reduce its synthesis. So the second substrate of our PKA protein kinase is the actual glycogen synthase. Here, phosphorylation downregulates the enzyme. We're downregulating the synthesis of glycogen. Then we said we had this third pathway here that is happening only in the liver, where we're dealing with this additional allosteric regulator of glycolysis and gluconeogenesis is fructose 2,6-bisphosphate, right? And remember, we said this is formed by a bifunctional enzyme that has one kinase domain and one phosphatase domain. And this too is a substrate of the protein kinase A. And it can, phosphorylation now here will do different things to the different domains. It will actually, phosphorylation will decrease the activity of the kinase. It will increase the activity of the phosphatase domain. That will, right, the phosphatase will dephosphorylate this fructose 2,6-bisphosphate. It will reduce the abundance of this allosteric regulator. As you're reducing the abundance of this regulator, it is an activator of our glycolytic kinase, the PFK1. So the lower the amount of the activator is, the less PFK1 is active. As an end result, you are reducing glycolysis. And we have said that's really important to have only in the liver, because the job of the liver in this situation, right, is to break down glycogen and to release free glucose into the bloodstream. So you want to reduce glycolysis as the opposing pathway direction, but you would only want to do so in the liver, because if you also were to do so in the muscle, you're reducing your capacity for making ATP in the muscle, you're reducing your capacity to move, and you don't want that, right? So this pathway is only glucagon triggered in the liver. So glucagon is coming in now, it's doing all these things, right? And remember, these pathways you should be aware of for the exam, right? We discussed them in detail in our lecture on metabolic regulation. Here we're just putting them into context into these different organismal conditions. So as your glucagon is now increasing, your blood glucose is dropping. What is happening now in the entire system now? So again, here in blue, we have the pathways following your carbohydrates. In orange, we're following your lipids. And then here in purple, we're following your amino acids. Right? And we just had said, right, glucagon is increasing. It will increase your glycogen breakdown to glucose 6-phosphate. So you can generate glucose that can be used by all tissues, specifically, right, you want it for the brain. In addition, right, you're running out of your carbohydrates now, so you will increase gluconeogenesis to make sure you have enough glucose for your brain and other tissues. To do gluconeogenesis, right, you need non-carbohydrate metabolites to do so. And so these will now come from your fats. So you're really tapping into your fat stores now. You're breaking down your fatty acids through beta oxidation. They either can go into gluconeogenesis, but not in animals. Remember that we can't do the de novo biosynthesis. We had to have the glyoxylate cycle for that. So what we are doing mostly is that actually the fatty acid breakdown will go into ketone bodies. And so they will actually now also be used, especially by the brain. But as, right, your blood glucose are gradually decreasing, your level of ketone body formation, ketogenesis, will gradually increase. And then in addition, you're also starting to actually break down your protein. And so that's broken down to amino acids. These go through your amino acid breakdown reactions, many of which can be used for either then the ETC to run or for gluconeogenesis to continue produce glucose that you need. But important here, right, you're starting to bypass the lack of carbohydrate resources by tapping into your fats and by tapping into your proteins, and you're increasing the production of glucose, especially for the brain. All right. Now assume you just keep going, right? You're so fascinated by watching BIS103 videos that you completely forgot eating all day. Now you're really, really hungry, right? So prolonged fasting, you're running now into sort of the level of almost starvation. What is happening now to your system, right? Glucagon will continue to increase. So these same effects that I just showed you for the intermediate fasting would still be in effect, and they would actually become more dramatic. So what is happening here now, right? You continue to do gluconeogenesis, and increasingly so, right? Because you need glucose for your brain. So you will push gluconeogenesis. You ran out of your carbohydrate resources a long time ago. So you're drawing on your TCA cycle intermediates largely to do so. Right? So you're drawing on your TCA cycle, you're drawing the metabolites away to do gluconeogenesis. At the same time, right, you are now increasing the use of your other energy metabolite resources, specifically your fats, right? These are your long-term storage. You increase fatty acid hydrolysis and beta oxidation. So you're generating a lot of acetyl-CoA that usually, right, would go into the TCA cycle for decarboxylation and to run the ETC. But because, right, you have been drawing metabolites away from your TCA cycle, the TCA cycle is less efficient now, which means you're starting to accumulate acetyl-CoA because you're increasing gluconeogenesis while at the same time increasing fatty acid breakdown. So you have a backlog here. The backlog causes an accumulation of acetyl-CoA. Now we're running into a cofactor issue, right? We have all our CoA trapped in our acetyl-CoA. And we had discussed, right, our fail-safe mechanism here is our ketogenesis. So combining acetyl-CoA molecules, releasing the cofactor, making ketone bodies, which also are now helpful, right, as an alternate energy metabolite resource, especially for your brain. Now, in addition to breaking down your fats, you're also in this state, right, as your carbohydrate resources, as your fatty acid resources, and your capacity to use fats because of this acetyl-CoA backlog are steadily decreasing. You're starting to eat more and more of your internal protein storage, right? So now you're really starting to use proteins for energy metabolism, so they go straight away into the TCA cycle for energy production. And keep in mind, right, you have to deal with your ammonium. So under these starvation conditions, you really, really are increasing now also your urea cycle. So in a way, like the activation of urea cycle is actually the same between the really well-fed state. You just have to deal with all the excess amino acids there. Here, it is increased urea cycle because you are increasingly tapping into your internal amino acid resources. So this is just to give you an idea of how are these pathways reciprocally regulated and working in concert with each other under these different conditions as you, for example, are moving through your days, are moving through your daily meals. And so this is just basically this concept in numbers. What this diagram shows you is actually the concentration of different energy metabolites across a period of almost 40 days. And so these are actual data from a pool of people who were willing to go through an experimental fasting for 40 days. And people have measured then these different energy metabolites to see what is actually happening. So a lot of the understanding of these reciprocally regulated pathways and how they work in concert under these conditions comes from these types of experiments. And so what you see, right, like here's glucose in blue again, right? Your blood glucose here is right here. So this falls very rapidly, right? Even over the first 2 days, your blood glucose is dropping. Then you kind of stop here. So your glycogen is kicking in really heavily. Glycogen is broken down to make sure that you can maintain blood glucose levels as best possible, right? Remember, usually you want to be at 5. We can use now glycogen breakdown over these first days, and then later, this is all gluconeogenesis essentially here, trying to maintain a bare minimum of the glucose concentration in your blood that you need, right? You see it actually barely is dropping past 4 or 3.5 here. So that's really gluconeogenesis kicking in over this long run here. At the same time, what you see, like free fatty acids are steadily increasing here and staying basically sort of a standard and steady resource of energy. But keep in mind your lipids, your fats, your fatty acids are sort of the really long-term storage of energy. So you're continuously using here your fatty acids over this entire period of time as a source of energy. But right, keep in mind, as we are doing this, we're increasing gluconeogenesis. We're starting to have our backlog of acetyl-CoA here, right? As we are sort of really more and more using fatty acids and beta oxidation, yielding acetyl-CoA. So at the same time, early on here already you see all our ketone bodies increasing, right? Here's acetone, here's your acetoacetate, and your beta-hydroxybutyrate. And you see, like, over time, these ketone bodies are really taking over as your energy resource over time. And this is because we cannot efficiently use acetyl-CoA from fatty acid breakdown because of this cofactor issue. And so this is essentially the same graph, just in numbers, to give you a bit of an idea of the time frames of this. They obviously are very variable because it depends on every person and how many resources you have internally. It depends on many factors, but it's just sort of as a ballpark, right? Your immediate resource, once all your blood glucose essentially has been used up, is your liver glycogen. But that really, within 2 days, you basically have used up your glycogen. So only in this first, essentially 1 to 2 days, you're using glycogen. But during this time, you're using almost exclusively glycogen as your energy resource there. Then sort of as time moves on, you're using your gluconeogenesis in the liver mostly from amino acids now, and that's increasingly high. So you're really using a lot of protein breakdown to meet your energy demand here. So it's almost 80% at the very beginning. Then you're tapping into your own proteins a lot. And so if you actually see the numbers here, but in the beginning, at day 3, you're using a lot of your protein. So this is really your major resource right now. It will actually at day 30 or 40. So as you are continuing to using up your protein resources for energy, it will drop to about only 1/5 of what it was before. So only about 2 times of your normal use, which really depends on how much protein resources you have internally. The reason here is, right, that initially here you would start to use a lot of your muscle protein, right? But as you're actually decreasing these resources, you're decreasing the use of protein from muscle because you're starting to tap into your essential proteins, right? You cannot use your heart muscle protein for energy, right? There are essential parts of protein that you cannot use. So as a mechanism of survival, as you are moving into this phase of starvation, as you fast for this time, you are actually dramatically reducing the use of proteins and an energy resource. And so this is really also then where your ketone bodies kick in. So it's the same time here again, right? Day 3, it's about 33% of your energy need are ketone bodies. And then as you're reducing using proteins, you're increasing the use of ketone bodies. A lot of this comes out of fatty acid breakdown, and the acetyl-CoA from that is used to make ketone bodies. So almost 3/4 of your energy demand that you have after 30 to 40 days will be met by ketone bodies. And we end at 40 here, depending on who you are and what your resources are, you cannot actually last much past 40 days without getting some food. Right, this concludes our section on metabolic integration. So I hope this was an enjoyable lecture to really put all of what you learned about our biochemical pathways, their opposing directions, their cooperation, and their reciprocal regulation together, and really look at how this works under different conditions in a human body.